review series on the organelles of the cell. Today, we're going to be talking about how eukaryotes communicate between the nucleus, the nucleolus, and the ribosomes. Now, the nucleus may just be the most important organelle in the cell. Inside the nucleus, we find DNA, RNA, and a whole bunch of proteins that are serving either structural or enzymatic roles. Now, the DNA normally is found in the form of chromatin. So if you follow along with the slide set, chromatin is this material inside the nucleus. Now, typically, you may have seen this chromatin when it's coiled up and condensed as chromosomes during cell division. But most of the life of the cell is not spent dividing, and so it's not coiled up. It's actually fairly loose in this region, and it has denser regions and less dense regions. And so to those regions, we give different names: euchromatin, heterochromatin. Now, within that nucleus also, we have a region of RNA, sometimes several regions, where RNA is very concentrated, and we've termed these the nucleoli. One would be a nucleolus or a nucleolus, and this is where the components, the subunits of ribosomes are made. The ribosomes aren't made whole in the nucleolus, but they're made in the large and small subunits that are then exported out the nuclear pore of the nuclear membrane into the cytoplasm. And then the third thing I said that we find in there is quite a lot of protein. This protein serves enzymatic roles, copying the DNA, also copying the DNA in a process called transcription and making mRNA, messenger RNA copies, that then can also leave out the nuclear pore. Here's another view of the nucleus. You can see in the slide where those different regions of chromatin are located. You can see the double membrane, that nuclear envelope that covers the outside of the nucleus. We've sliced through, so you're looking at it like a circle, but it's really more spherical. And then, of course, it has all these holes in it. So in many ways, it kind of resembles a wiffle ball or a golf ball with all the dimples and holes on the outside. You'll notice also in this slide that the rough endoplasmic reticulum is continuous with the nuclear envelope. Here's a close-up view, an electron micrograph showing the nuclear envelope with the associated ribosomes, and then here's the inside of the nucleus. The nuclear pore, close-up of that, and so you can see how there's this inside space to the nuclear envelope between the two parts that are membrane. This inside space in organelles is termed a lumen, L-U-M-E-N. And then the nuclear pore complexes. If we looked at the surface of the nucleus, we would see those pores at regular spaces across the surface of the nucleus. Pores that allow the ribosomal subunits to pass out of the nucleus, pores that allow mRNA to pass out of the nucleus, and then, of course, allow the proteins that are needed to pass into the nucleus from where they're made at other places in the cell. The whole process of protein synthesis looks somewhat like this. Within the nucleus, here above the green curved line that represents the nuclear envelope, we have the process of copying DNA for when we might wish to make a copy of the whole cell, but also on a regular basis when the cell is not going through reproduction, we transcribe certain sections of the DNA known as genes by making little messenger RNA or mRNA copies. These copies can then go out the nuclear pore into the cytoplasm. Now, out here in the cytoplasm, these copies are then translated by a ribosome into a protein. So all of the genetic information stored by a DNA ultimately becomes active, communicates with the rest of the cell by being translated into proteins. And then those proteins, whether they be structural or enzymatic or whatever role they're going to function, maybe hormonal, then go off to do their job. Now, ribosomes come in two types. They come in the free type, that is unattached in the cytoplasm, and when those ribosomes read mRNA and translate it into a protein, the proteins are for the cell's own use, for use in the cytoplasm. But ribosomes also might read a signal sequence on the end of that mRNA, and because of the way they've read that mRNA, they know to go ahead and attach themselves to the rough endoplasmic reticulum. And when they do that, then the protein that they've made is actually made into the inside the lumen of the rough endoplasmic reticulum, and then it's used for transport to the Golgi apparatus, the rest of the membrane system, or for use in making an organelle, or perhaps even for export from the cell. You can see that once it gets inside here, this little enzyme, signal peptidase, cuts off that signal once the ribosome has bound with the rough endoplasmic reticulum. Well, talking about ribosomes bound to rough endoplasmic reticulum really leads us into our next topic, which is the endomembrane system. But that's for another video. So once again, in summary, the organelles that begin communicating in the cell start with the nucleus, where the DNA, recipes for proteins, are held in the form of those gene sequences on the DNA that then get transcribed into mRNA, leave the nucleus by the nuclear pore, get translated into proteins by the ribosomes out in the cytoplasm, both free and bound. The ribosomes themselves were made in two components, the large ribosomal subunit and the small ribosomal subunit, which were made in the nucleus. They're also made out of RNA material, ribosomal RNA, or rRNA. And that brings us to the end of our discussion of the nucleus, the nucleolus, and the ribosomes. Thanks for watching.